Welcome to this video on genetic engineering. In this lesson, we'll explore one of the most revolutionary technologies in modern biotechnology. Genetic engineering allows scientists to directly modify the DNA of living organisms, opening up incredible possibilities in medicine, agriculture, and research. Let's dive into how this fascinating technology works and why it matters. Genetic engineering is the deliberate modification of an organism's genome by directly manipulating its DNA to introduce, remove, or alter specific genes. Think of it as precision editing of the instruction manual that controls how organisms develop and function. Unlike traditional breeding, which takes many generations, genetic engineering allows scientists to make targeted changes quickly and accurately. This gives us unprecedented control over the traits and characteristics of living organisms. Genetic engineering operates on four key principles. First is precision, which means scientists can target specific genes with molecular accuracy rather than making random changes. Second is heritability, meaning the genetic modifications pass on to future generations through reproduction. Third, these changes are controlled and intentional, using advanced biotechnology tools rather than leaving things to chance. Finally, the modifications are functional, meaning they actually alter the traits and characteristics of the organism in meaningful ways. The genetic engineering process follows three main steps. First, scientists identify and isolate the target gene they want to work with. This could be a gene that produces a useful protein or one that needs to be modified. Second, they insert this gene into the host organism's DNA using specialized molecular tools. Finally, the new genetic information is expressed, meaning the organism starts producing the desired protein or displaying the new trait. This process requires precision and careful control at every stage. Genetic engineering relies on several essential tools. Restriction enzymes act like molecular scissors, cutting DNA at specific sequences. DNA ligase works like molecular glue, joining DNA fragments together. Vectors, such as plasmids, are used to carry genes into cells. Plasmids are small circular DNA molecules that can replicate independently. PCR, or polymerase chain reaction, amplifies DNA sequences to create millions of copies. Finally, CRISPR-Cas9 represents the cutting edge of gene editing technology, allowing precise modifications with unprecedented accuracy. CRISPR-Cas9 is a revolutionary gene editing technology that has transformed genetic engineering. It works like molecular scissors, allowing scientists to cut and edit DNA at precisely targeted locations. The system uses a guide RNA molecule that directs the Cas9 enzyme to a specific DNA sequence. Once there, the enzyme cuts the DNA, and scientists can remove, replace, or insert genetic material. This technology is faster, cheaper, and more accurate than previous methods, making genetic engineering more accessible than ever before. Genetic engineering has wide-ranging applications across multiple fields. In medicine, it's used to produce life-saving drugs like insulin and to develop vaccines and gene therapies for treating diseases. In agriculture, scientists create crops that resist pests and produce higher yields, helping to address food security. Research laboratories use genetic engineering to study how genes function and to understand disease mechanisms. In industry, engineered organisms produce useful enzymes and biofuels more efficiently than traditional methods, offering sustainable alternatives to conventional production. Let's look at some real-world examples of genetic engineering in action. Bt corn contains a gene from bacteria that produces a protein toxic to certain insects, reducing the need for chemical pesticides. Human insulin is now produced by bacteria that have been engineered with the human insulin gene, providing a reliable supply for diabetes patients. GloFish are zebrafish modified with genes from jellyfish that make them glow under ultraviolet light. These examples show how genetic engineering is already part of our everyday lives. Genetic engineering offers tremendous benefits, but also raises important ethical concerns. On the positive side, it enables disease treatment and prevention, enhances food security through improved crops, provides environmental solutions like biodegradable plastics, and advances scientific knowledge. However, we must consider potential unintended consequences of genetic modifications, possible ecological impacts on ecosystems, questions about ethical boundaries in modifying life, and issues of access and equity in who benefits from these technologies. Balancing these benefits and concerns is crucial as we move forward. To summarize, genetic engineering is a powerful biotechnology tool that enables precise modification of genomes. It has transformed medicine by producing life-saving treatments, revolutionized agriculture by creating improved crops, and advanced scientific research in countless ways. However, this power comes with responsibility. As we continue to develop and apply genetic engineering technologies, we must carefully consider the ethical implications and ensure these tools are used for the benefit of all. Understanding genetic engineering is essential for navigating the future of biotechnology and making informed decisions about its applications.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.